This is Group 19. Let me tell you about VDJ recombination. Before getting into the main topic, let's review what fast antibodies are made of. The basic structure of an antibody is Y-shaped and consists of two heavy chains and two light chains. These changes each have a variable region and a constant region, and the amino acid sequences of variable regions differ depending on the antibody. Antibodies with this structure bind to specific antigens and eliminate pathogens. We have some questions about this system. The question is, although one antibody can recognize one specific antigen, how can our bodies recognize contrast antigen? The answer can be explained by a process called VDJ recombination. VDJ recombination is the process of randomly combining various gene fragments in antibodies to increase the number of types of antibodies. The number of types of antibodies increased through this process is 1 billion.
In answer to the previous question, VDJ recombination increases the number of types of antibodies, making it possible to recognize almost pathogens. Additionally, T-cell receptor is similar to antibodies and can recognize various antigens by changing their TCR. VDJ recombination occurs when B cells and T cells are created. B cells occur in the bone marrow, and T cells occur in the thymus cortex.
When D M cells, precursor cells of T cells, become DP cell, and pre-V cell become pro-V cell, recombination occurs through the action of the enzyme LAG1 and LAG2, respectively.
As mentioned earlier, there are variable regions in the heavy chain and light chain of antibodies, and the gene for the heavy chain variable region of antibodies is found in three types of gene regions: V, D, and J, and the gene for the light chain variable regions is divided into two parts, V and J. One gene fragment was selected from each part, 10,530 patterns for the heavy chain. Similarly, 200 for the light chain, in 2,106,000 gene types. In addition, more than 1 billion types of antibodies are produced due to genetic mutations.
Also, in explaining VDJ recombination, we have to mention 12/23 rule. Each segment of VDJ has RSS next to it, and RSS contains the sequences 7-MAR and 9-MAR. There is always a 12 base pair or 23 base pair spacer sequence between these sequences. 12/23 rule allows recombination only between RSS with 12 base pair and 23 base pair spacer sequences, which ensure correct differentiation. Incorrect recombination does not occur. Enzymes called LAG1 and LAG2 protein binds to the RSS and cleaves the DNA double strand, resulting in VDJ recombination.